Hello friends, myself Professor Seema Nagdeve, Assistant Professor and Head of the Department Botany, Mahatma Gandhi College, Armory District, Gadchiroli. As per CBCS syllabus of Botany of Gondwana University, Gadchiroli, in B.Sc. second year semester 4, 4 units are included. In first unit, there is a study of ultrastructure and function of typical plant cell wall, plasma membrane, general structure of nucleus, mitochondria, plastids, endoplasmic reticulum, Golgi bodies, vacuole, lysosome, peroxisome, and gliocisome. It also includes cell division, in which mitosis, meiosis with respect to plant cells is included. In DNA, there is a structure and the replication of DNA, and also study of plant tissue culture, in which concept of totipotency, steps of plant tissue culture from explant to whole plant regeneration. This is about the introduction of unit 1 of paper 1. Now, course outcomes of this unit 1: to know the ultrastructure and function of typical plant cell, to study the detailed account on cell wall and plasma membrane, to study the general structure of nucleus, mitochondria, plastids, and endoplasmic reticulum, to study the general structure of Golgi complex, vacuole, lysosomes, peroxisome, and gliocisome, to study cell division, that is mitosis, meiosis with respect to plant cells, to know the structure and replication of DNA, to study about plant tissue culture, in which concept of totipotency, steps of plant tissue culture from explant to whole plant regeneration is included. Now about unit 2. In this unit, there is a law of inheritance of Mendel, in which 3 laws are included. Number first is law of dominance, second one law of segregation, that is law of purity of gametes, and third one law of independent assortment. It also includes backcross and test cross. Interaction of genes with reference to plants includes allelic interaction, in which incomplete dominance, and non-allelic interaction, in which complementary genes, the ratio of complementary genes is 9:7, and also supplementary genes, that is 9:3:4 is included. In extranuclear genome, there is a structure and functions of mitochondrial and plastid DNA. Now, this is about introduction of unit 2. Now, the course outcomes of this unit: to study the detailed account of Mendel's laws of inheritance, to study the gene interaction with reference to plants, to study the allelic and non-allelic gene interaction, to study complementary and supplementary genes, to know the extranuclear genome, that is the structure and function of mitochondrial and plastid DNA. Now about unit 3. In unit 3, there is a study of linkage, in which definition, gene theory of Morgan, types of linkage, that is complete and incomplete linkage with its significance is included. In crossing over, there is a definition, theories, that is breakage and reunion, and copy choice theory with its significance is included. In variation in chromosome number, polyploidy, 2 types of polyploidy, that is autopolyploidy and allopolyploidy, as well as aneuploidy, which includes nullisomy, monosomy, trisomy, and tetrasomy with its significance. Now about structural changes in chromosome, that is chromosomal aberration, in which deletion and deficiency, duplication, inversion, and translocation is included. In mutation, there is a definition, types of mutation, that is spontaneous and induced, substitution and frame shift mutation, types of mutagens, that is physical and chemical mutagens, application of induced mutation in crop improvement. Now the course outcomes of this unit 3: To know the linkage, that is definition, gene theory of Morgan, types of linkage, complete and incomplete linkage with its significance. To study the detailed account of crossing over, to study the variation in chromosome number, to know the structural changes in chromosome, to study the mutation, that is induced mutation and natural mutation. This is about the course outcomes of Unit 3. Now about Unit 4, genetic engineering. It includes tools and techniques of recombinant DNA technology. In cloning vectors, there is a study of plasmids, bacteriophages, lambda phage, and Agrobacterium. Also, it includes restriction enzymes and ligases, genomic and complementary DNA libraries, protein synthesis in which transcription and translation is studied. In jumping genes, there is AC and DC elements in maize. In regulation of gene action in prokaryotic, there is a study of lac operon concept. Now about course outcomes of this unit. To know the genetic engineering, to study detailed account of tools and techniques of recombinant DNA technology, to study the cloning vectors, to study the restriction enzymes and ligases, to study genomic and complementary DNA, to know jumping genes, and to understand regulation of gene action in prokaryotes, lac operon concept. Now the summary of these course outcomes of paper 1, semester 4. Students learn about structure of typical plant cell and cell organelles, which contains mitochondria, chloroplast, endoplasmic reticulum, vacuoles, ribosomes, and Golgi bodies. Students understand the various steps of cell division, that is mitosis and meiosis, to understand tissue culture techniques. They also learn more about mendelism. They also knows about complementary and supplementary factors. They learn about plastids and mitochondrial DNA. They learn about mechanism of linkages and crossing over. To know more about polyploidy, mutation, chromosomal aberration, they learn mechanism of protein synthesis and also learn more about Agrobacterium, plasmid, T4 bacteriophage, jumping genes, and lac operon model. Thank you.